Hey guys, welcome back. Today's topic is about the plant hormones. Here, coordination in plants. The movement of the sensitive plant in response to touch is very quick. The movement of sunflowers in response to day or night, on the other hand, is quite slow. So, in this case, we find very quick movement, and here we find a quite slow movement. Here, growth related movements of plants will be even slower. See the difference. The movements in different plants in different situations, they are with different speed. You can see here the sensitive plant movement is very quick and the sunflower movement is quite slow, and the growth related movement of plants will be even slower. See the comparison. Okay, so controlled movements can be either slow or fast. So this is what happens. Now if we move to the next here, if fast responses to stimuli are to be made, information transfer must happen very quickly. So here, if fast responses, in order to achieve fast responses to stimuli, okay, what is needed? Information transfer must happen very quickly, and electrical impulses are an excellent means for this. Okay, they can transmit very fastly. They can be transmitted very fastly in the body from one part to another part, and electrical impulses are an excellent means for the transfer of information very quickly. Here there are two limitations to the use of electrical impulses. Okay, and this is one of the very important question for board exam. Okay, so this question can be asked for 2 marks. Here the question is, what are the limitations of electrical impulses? Here, they will reach only those cells that are connected by nervous tissue. So here only those cells which are connected by nervous tissue will receive the electrical impulses, and the cell will take some time to reset the mechanisms before it can generate and transmit a new impulse. So this is another limitation. So these are the limitations of electrical impulses. First, they will reach only those cells that are connected by nervous tissue, and the second, cell will take some time to reset the mechanisms before it can generate and transmit a new impulse. Okay, so these are the limitations of electrical impulses. In other words, cells cannot continually create and transmit the electrical impulses. Okay, so no wonder that most multicellular organisms use another means of communication, and that another means of communication between cells is nothing but the chemical communication. Okay, so this is one more mode of communication seen between the cells. As there are limitations with electrical impulses, the another mode of communication is chemical communication. Let's discuss about this in detail. Here, if instead of generating an electrical impulse, stimulated cells release a chemical compound, this compound would diffuse all around the original cell. Let's suppose this is a cell. Okay, and now it is stimulated. If the stimulated cells release a chemical, let's suppose this is a chemical, and this chemical would diffuse all around the original cell. It will diffuse all around the original cell. If other cells around have means to detect this compound, using special molecules on their surfaces, then they would be able to recognize information and even transmit it. So this is how the chemical compounds, here we are going to discuss about these chemical compounds in detail. They are nothing but the hormones. So the stimulated cells, if they are releasing these hormones, they diffuse all around the cell, and the nearby cells, if they have some special molecules to detect these chemicals, then obviously it would recognize the information and even transmit the information to other cells. So this is how chemical communication between cells work. Okay, now the continuation part. This will be slower, of course, but it can potentially reach all cells of the body. I repeat this once again, very important statement. This chemical communication will be slower, of course, but it can potentially reach all cells of the body, and it can be done steadily and persistently. So this is what happens with chemical communication. It happens steadily and persistently. So this is what needed in living organisms for continuous responses as per the requirement. Okay, so these compounds are called hormones. They are used by multicellular organisms for control and coordination, show a great deal of diversity. So because of hormones, the multicellular organisms, okay, they show very appropriate responses, and here there is a great deal of diversity in showing the responses. Okay, so this is about introduction part of the chemical communication, that is the hormones play important role. Now moving to the next, actually what are these hormones, what are they also called, and how do they affect different functions in plant body? Let's discuss in detail. Here we use the words plant hormones, or we can also call phytohormones. These are the other words. Okay, so plant hormones are also called phytohormones or plant growth regulators. In short, they are called PGRs. Okay, these are small, these are simple molecules of diverse chemical composition. So plant growth regulators are smaller and simpler molecules of diverse chemical composition, and the PGRs can be broadly divided into two groups based on their functions in a living plant body. So based on their functions, they are broadly divided into two. So two groups of plant hormones. Let's discuss what are those two groups. Here, one group of PGRs are involved in growth promoting activities. So these are growth promoting hormones. These are involved in growth promoting activities such as cell division, cell enlargement, then pattern formation, then tropic growth, flowering, fruiting, seed formation. So like this, there are so many growth promoting activities. So this one group of PGRs, plant growth regulators, they are involved in growth promoting activities. So what are the growth promoting activities? Here the answer is the activities like cell division, cell enlargement, pattern formation, tropic growth. It means directional movement shown by the plants. Okay, and flowering, fruiting, seed formation, all these are growth promoting activities, and this group of plant growth regulators are involved in all these activities. Okay, so these are also called plant growth promoters, and examples for this group of hormones: Auxins, Gibberellins, and Cytokinins. So these hormones are plant growth promoters, plant growth regulators. They are growth promoting hormones. Now, moving to the next, here the PGRs of the other group play an important role in plant responses to wounds and stresses of biotic and abiotic origin. So here the biotic is related to the living organisms, the parasites, the pathogens, all these. They are biotic stresses. And abiotic origin, the climatic conditions. Okay, so both towards both these conditions, the plants will show responses, and certain hormones are involved in that. So the other group of hormones play important role in plant responses to wounds and stresses to biotic and abiotic origin. And here they are also involved in various growth inhibiting activities such as dormancy and abscission. Dormancy is inactive condition, okay, and abscission is, for example, falling of leaves, falling of fruits, like this. Okay, these are abscission activities. So here the hormones of this group, they are involved in growth inhibiting activities such as dormancy or abscission. And here the PGR, abscisic acid, belongs to this group. Abscission, abscisic acid, there is a similar word. You should remember this in order to say its function. Yes, abscisic acid function is abscission. Abscission is nothing but the falling of leaves, falling of immature unripened fruits, like this. Okay, so this is about, okay, the other group of plant hormone which are involved in growth inhibiting activities. Okay, one more, the gaseous PGR. Okay, the gaseous plant growth regulator could be fit in either group. It will fit in either group. It means it is both growth promoting activities as well as growth inhibiting activities. In both these activities, this gaseous PGR, it is called ethylene. Okay, so it is involved in both the functions. So dual role, both growth promoting activities and growth inhibiting activities. So which hormone is involved in both growth promoting activities as well as growth inhibiting activities? What is your answer? The answer is ethylene. Okay, now let's discuss about these plant hormones. Few more details. Different plant hormones help to coordinate growth, development, response to the environment. Okay, they are synthesized at places away from where they act. Very important statement. Okay, so here I repeat this once again. These plant hormones are synthesized at places away from where they act and simply diffuse to the area of action. Okay, so at far places they are synthesized and they diffuse to the place of action. This is what happens in case of plant hormones. Now let's discuss about these plant hormones in detail. Here one of the plant hormone is auxin. Okay, so here auxins. Auxins are generally produced by the growing apices of stems and roots from where they migrate to the regions of their action. So just now I mentioned one of the important statement, plant hormones are synthesized at the places far away from their place of action. So the similar aspect here applicable. So you can see here auxins are produced by the growing apices, apex parts, okay, like apex of the stem, apex of the root, tip regions. That is what here the meaning is. Okay, so growing apices of stems and roots from where they migrate to the regions of their action. It means they diffuse, okay, and they reach the site of their action. Okay, now these auxins help to initiate rooting in stem cutting. So this is one of the very important function performed by auxins. They initiate rooting in stem cuttings. Okay, so some of the plants, they are growing by vegetative propagation. Stem cuttings are used. So in such cases, initiating root is one of the function performed by auxin. So auxins help to initiate rooting in stem cuttings. Okay, in most higher plants, the growing apical bud inhibits the growth of the lateral buds, a phenomenon. This is what we call apical dominance. Very important question for 1 mark. What is apical dominance? Apex means tip region. Apex means tip region. Here the shoot tip grows faster than the lateral branches. So this is what the meaning here is. Okay, the apical buds inhibits the growth of the lateral buds, and this phenomenon is called apical dominance, and auxins are responsible for apical dominance. If you look here in this picture, this is apex, apical bud. Okay, and you can see here, okay, because of the auxins, because of the plant hormones auxins, this apical bud, this is how it looks like. It is growing faster, but when it is detached, when the apical bud is detached, you can see here the lateral branch. Okay, lateral branch is growing. It is growing in the absence of apical bud. Here the apical bud is present, so it will grow. Here the apical bud is detached, so obviously the lateral branch, a new branch is grown, and it will continue. So this is what happens. Auxins play important role in apical dominance. It means apical buds inhibit the growth of lateral buds. In the presence of apical bud, lateral buds do not grow, axillary buds do not grow. So that is what the meaning is. Okay, now the continuation part. Auxins help to prevent fruit and leaf drop. Okay, so they are preventing the fruit and leaf drop at early stages, but promote the abscission of older mature leaves and fruits. So this is one of the very important statement. Okay, what the auxins are doing, there is a clarity here. They help to prevent fruit and leaf drop at early stages, but promote abscission of older mature leaves and fruits. Okay, so this is advantageous for the farmers. Okay, now auxins also induce parthenocarpy. For example, in tomatoes, parthenocarpy is nothing but seedless fruits. Okay, seedless fruits are formed by the action of these auxins. So auxins also induce parthenocarpy. It is nothing but the formation of seedless fruits. They are widely used as herbicides. Herbicides are the chemicals which kill herbs. Okay, so unwanted herbs that are growing in the field, they are killed by auxins. So they are widely used as herbicides. For example, 2,4-D is widely used to kill dicotyledonous weeds. Okay, does not affect mature monocotyledonous plants. So these herbicides, which are nothing but the auxins, they do not affect mature monocotyledonous plants, but they kill dicotyledonous weeds present in the field. So this is how auxins are helpful for the farmers in agriculture fields. They destroy dicot weeds in the field. Okay, so finally the functions of auxins are like this. Okay, they are initiating rooting in stem cuttings. Okay, in most higher plants, growing of apical buds inhibits the growth of lateral buds. This is apical dominance because of auxins, and then they help to prevent the fruit and leaf drop at early stages, but promote the abscission of older mature leaves and fruits. Okay, so here auxins also induce parthenocarpy, and they are widely used as herbicides. These are the functions of plant growth regulators auxins. Now let's discuss about here. When growing plants detect light, a hormone called auxin is synthesized at shoot tip, helps the cells to grow longer. One of the very important mechanism that we are discussing now. This is very important question for exams as well. Okay, so when light is coming from one side of the plant, auxin diffuses towards the shady side of the shoot. This concentration of auxin stimulates the cells to grow longer on the side of the shoot which is away from light, and thus the plant appears to bend towards light. Let's suppose from this side the light is coming and here is a plant. Let's suppose now this part is receiving light and this side is shade. Okay, this side is light. So as per the discussion that we had till now, where is the hormone synthesized? So hormones are synthesized at the tip regions. So here now when growing plants detect light, a hormone called auxin synthesized at the shoot tip helps the cells to grow longer. Okay, when light is coming from one side of the plant, the auxin diffuses towards the shady. So the auxin here, it will diffuse towards the shady side. So this side cells will grow longer when compared with the region that is receiving the light. So obviously these cells grow longer and they bend towards light like this. So this bending movement in plants is because of the plant hormone auxin. Okay, so this is one of the very important question for board exam and also for competitive exams. Okay, so here auxins are responsible for bending movement, and this bending movement occurs because of unilateral growth. That is, one side we find more growth, other side we find less growth. This difference in growth is because of the difference in concentration of the plant hormone, that is auxin. Okay, so this is all about the bending movement because of auxins. Now moving to the next. Here after this, gibberellins. Gibberellins help in the growth of stem. So stem elongation, okay, they are responsible for stem elongation. If the dwarf plants, okay, if we apply gibberellins to dwarf plants, then stem elongation can be seen. So they play very important role in stem elongation. Their ability to cause an increase in length of axis is used to increase length of grape stalks. So this is how they are increasing the length of grape stalks. Okay, and they are also delay senescence. Senescence is nothing but the falling. Okay, so here gibberellins delay the process of senescence. Thus the fruits can be left on the tree longer so as to extend the market period. So here the meaning is the young fruits, the smaller fruits do not fall. That is what delaying the senescence process. Okay, so gibberellins delay senescence so that the fruits can be left on the tree a longer time. Okay, now the continuation. Gibberellins also promote bolting. This is one important word. It is nothing but internode elongation just prior to flowering. Okay, so internode, let's suppose this is the stem. This is one node. This is another node. Between these two is internode. Okay, so gibberellins are responsible for internodal elongation. So you can see this is the length of the internode. Here it is changed, increase. So this is what we call bolting. Okay, so gibberellins also promote bolting, that is internode elongation just prior to flowering. In beet, cabbage and many plants, with rosette habit. So this is one important function of gibberellins. Spraying sugarcane crop with gibberellins increase the length of stem, thus increasing the yield. So stem elongation occurs even in sugarcane crop because of this. Okay, so the main function of gibberellins is increasing the internodal length. Okay, so increasing the stem length, that is what the function of gibberellins. Now moving to the next, here cytokinin. Cytokinins promote cell division. This is one of the very important question. Okay, so which hormone is responsible for cell division? The answer is cytokinin, and it is natural then that they are present in greater concentration in areas of rapid cell division. Where do we find rapid cell division? We find it in fruits and seeds. Okay, so obviously in such places, okay, in fruits and seeds, which hormone will be present in higher concentration? The hormone will be cytokinins. Natural cytokinins are synthesized in regions where rapid cell division occurs. For example, root apices, developing shoot buds, young fruits. So root apices means tip regions, root tips. Okay, and then developing shoot buds, then young fruits. These places, they have higher concentration of cytokinin, and these regions show rapid cell division. Okay, now the continuation part of this, it helps to produce new leaves, chloroplasts in leaves, lateral shoot growth, and adventitious shoot formation. So these are some other functions of cytokinins. So new leaves are formed because of cytokinins, many chloroplasts are formed in leaves, and lateral shoot growth is because of cytokinins, and adventitious shoot formation is because of cytokinins. They help overcome the apical dominance. Apical dominance is the function of auxins, and cytokinins are opposite to that. We use the word antagonistic. Okay, so here apical dominance is opposite here, lateral dominance. Apical dominance, tip regions grow faster. This is the function of auxins. Okay, but here cytokinins, they are responsible for lateral shoot development, and this is what lateral dominance. Okay, so cytokinins are responsible for this. So here we use the word antagonistic, means they are opposite in their function with respect to the growth, apical dominance or the lateral shoot growth. So this is about the cytokinins. They promote nutrient mobilization, which helps in the delay of leaf senescence. So if nutrients are mobilized, if they are supplied as per the requirement, then obviously the leaf falling will be prevented. So here delay of leaf senescence is because of cytokinins, because they are promoting nutrient mobilization and help in delaying the leaf senescence, that is nothing but the falling of leaves. Okay, after this, the next one, ethylene. It is synthesized in large amount by tissues undergoing senescence and ripening of fruits. Okay, so very important question. So which hormone is responsible for fruit ripening? The answer is ethylene. Okay, ethylene promotes senescence, abscission of plant organs, especially leaves and flowers. So here in the beginning we discussed, this is a gaseous plant growth regulator, and this hormone is having a dual role. What are the dual roles? Growth promoting as well as growth inhibiting. Promote the growth and inhibit the growth. So both the functions are performed by ethylene. So here ripening of fruits is a type of promoting activity, and you can see here promotes senescence and abscission. This is inhibiting activity. So this is how it is involved in both the activities. ethylene is highly effective in fruit ripening. Okay, so very important question for exams. Ethylene breaks seed and bud dormancy, initiates germination in peanut seeds, sprouting of potato tubers. So here it is positive. It means it is promoting activity. What it is doing, it is initiating germination in peanut seeds and sprouting of potato tubers. So this is growth promoting activity. Okay, ethylene also promotes root growth and root hair formation, thus helping the plants to increase their absorption surface area. So this is also positive. It is promoting root growth. It means growth promoting activity. And root hair formation, obviously these are growth promoting activities. They help the plants to increase their absorption surface. So these are growth promoting activities. Now here moving to one more plant hormone, that is abscisic acid. Abscisic acid is one example of a hormone which inhibits growth. This is perfectly inhibiting hormone. Okay, in the beginning we discussed growth promoting hormones, auxins, gibberellins, cytokinins. Then we discussed about ethylene, which has a dual role, both growth promoting activity as well as growth inhibiting activity. Now we are discussing about abscisic acid. This is growth inhibiting hormone. Okay, its effects include wilting of leaves, okay, folding of leaves, wilting of leaves, and it stops, okay, growth. Okay, it stops growth. Let's discuss with few example. Abscisic acid, short form is ABA, inhibits seed germination. See, seed germination is actually a growth promotion, but what it is doing, it is inhibiting seed germination. Abscisic acid stimulates the closure of stomata and increases the tolerance of plants to various kinds of stresses. Therefore it is called stress hormone. This is very important question for exams. So which plant growth regulator is also called stress hormone? What is your answer? The answer is ABA, that is abscisic acid. So abscisic acid is a stress hormone in plants because it stimulates the closure of stomata and increases the tolerance of plants to various kinds of stresses. Therefore it is a stress hormone in plants. By inducing dormancy, ABA helps seeds to withstand desiccation and other factors unfavorable for growth. So here desiccation is water is lost, extreme water loss from the cells, that is desiccation. So by inducing dormancy, abscisic acid helps seeds to withstand desiccation. So this abscisic acid is inducing the dormancy in seeds. So seed dormancy is because of this. So it is growth inhibiting activity and other factors unfavorable for growth. So this is how it is inducing the plants to tolerate the unfavorable condition. So it is a stress hormone. So these are the different plant growth regulators. Among the 5 different types of plant growth regulators, auxins, gibberellins, cytokinins are growth promoting hormones, and ethylene is performing the dual role, both growth promoting activities as well as growth inhibiting activity, and finally abscisic acid is having the growth inhibiting activity. So this is all about today's topic. If you are new to my channel, subscribe for more sessions. If you liked it, share it with your friends. I'll be back with more sessions. Have a great day.